Hey everyone, welcome to our review of genetics. This is a very basic overview of genetics. We went a lot more in depth in class, but this is going to give you kind of some of those basic ideas that we talked about with genetics, and hopefully that will remind you of more that we talked about genetics. So, first, let's talk about the father of modern genetics. His picture is down here. His name is Gregor Mendel. He was a monk, he lived in a monastery, and he made observations about pea plants and the different traits and how they were passed on from one generation of pea plants onto the next generation of pea plants. And what he noticed is that sometimes a certain trait would show up in one generation and then skip a generation, not show up in their offspring, and then show up again later. And that was kind of interesting to him. And he started to realize that traits could be passed, but that sometimes they could be masked by another trait and not seen again until a later generation. And we'll talk about that. That's the idea of dominant versus recessive. But first, let's talk about some different vocab you'll want to know. Genotype versus phenotype is our first kind of comparison side by side vocabulary. The genotype is the type of genes that a certain organism has, an individual has. You show them as alleles, so they have two copies of each gene, one from their mother and the other from their father. And if they got the dominant trait from each parent, they would have a dominant genotype, big B, big B. If they got a dominant one from their mother and a recessive one from their father, it would be big B, little b. Or there could be another genotype, little b, little b. That would be if they got recessive from each parent. But the genotype basically shows which copy of the genes did a person get from their mother or father. Next, we have phenotype, and that would be the physical appearance, meaning, okay, they're tall or they're short. So, how they actually physically appear, or could possibly be something that they're able to do, such as curl the tongue or not curl the tongue. So, genotype, which gene did they get, or which copy of the gene did they get, the dominant or the recessive one? And phenotype is how do they actually look or what are they able to do? The next comparison of vocab would be dominant and recessive. The dominant trait is the trait that is usually expressed. We use a capital letter if we're talking about genotype, like a big B, to show the dominant genotype. And then also we have recessive, which means that's the one that's only going to be shown when there's not a dominant allele. We use a lowercase letter like a little b to show recessive, and that trait, that phenotype of a recessive trait is only going to show up if it's a lowercase, like a little b for the genotype. All right, next up we have a comparison of homozygous and heterozygous. Homozygous means two of the same allele. Another word for that is purebred. So if you say you have a purebred mother and a purebred father, that means they're both homozygous for whichever traits they have. Heterozygous means that you have two different alleles, usually one dominant and one recessive, and that is another word for hybrid. So if you say you have two hybrid parents, you have two heterozygous parents. That means that they're like big B, little b, two different types of the allele. Next up, we have autosomal versus sex linked. If you have an autosomal trait, you have a trait that is on one of your genes, one of your chromosomes that is the first, second, third, fourth, fifth, all the way up to 22nd chromosome. So if it's on one of those pairs 1 through 22, that's an autosomal trait. A lot of times you might see dominant and recessive within autosomal traits. So in pedigrees, which are these little diagrams that show genetic traits through several generations of a family, you could see a certain autosomal trait usually skip generations. The other type of trait you might see is a sex linked trait, and those are the ones that are on the 23rd pair of chromosomes, in other words, your X or Y chromosomes. And with sex linked traits, the males are actually more likely to suffer because of sex linked traits. We'll talk about that in just a second. Last side by side comparison is our two laws that we talked about with genetics, the law of independent assortment and the law of segregation. Now, those two laws end up resulting in offspring that are genetically different from their parents. So, in order to make offspring who are different from their parents, you rely on those two laws, independent assortment and segregation, and also crossing over, which we talked about previously. So, with the law of independent assortment, that basically means that if you inherit one gene, for example, the gene for widow's peak, it has no impact on whether or not you're going to inherit another gene, like the gene to curl your tongue. Basically, traits are separated. The genes for traits are independent of each other and they will be inherited separately of each other. So, just because you get a widow's peak does not mean you're going to be able to curl your tongue. The law of segregation means it's up to chance which copy of your parents' genes are going to be passed to you during meiosis. So, in other words, when the cell splits, it's completely up to chance which chromosome goes which way. And so, therefore, in the law of segregation, it's up to chance which copy of the gene your parents are going to pass you. So, let's say your parent was heterozygous or hybrid, it's up to chance whether they will pass you their dominant gene or their recessive gene. All right, speaking of passing genes, we use these Punnett square diagrams to show the possible gene combinations that could result from a genetic cross, meaning that two parents get together and have an offspring. This is called a monohybrid cross. Note the word hybrid. That means that both parents are heterozygous. So see heterozygous here and heterozygous here. Let's say this is the mother. She had a big A and a little a, and because of law of segregation, it's up to chance which one she'd pass to her offspring. Same with the father here. But when they cross, you can see in this diagram the possible offspring that two heterozygous parents might have. If you map out and fill in the Punnett square chart, you'll see that this offspring could be big A, this offspring could be big A, little a, this one could be big A, little a, and this one could be little a, little a. The way that I get those is by taking what's in the column and what's in the row and combining them. So in this big A, little a heterozygous offspring, it's because a big A from mom, a little a from dad. And let's talk a little bit about these offspring. This offspring is homozygous. They have both big and big. They're two of the same allele, homozygous and also dominant. This offspring is heterozygous. They have one big and one little a. This offspring is heterozygous, and both of these heterozygous offspring are going to show the dominant trait because if you have at least one copy of the dominant allele, you will show the dominant trait. Finally, we have little a, and that is homozygous because they're both the same, but it is recessive. Homozygous recessive is going to be shown there. So, if let's say these traits were for being able to curl your tongue, and being able to curl the tongue is a dominant trait, so the mom could curl her tongue because she was heterozygous, the dad could curl his tongue because he's heterozygous, and now 75% or 3 out of 4 offspring will be able to curl their tongue because they got at least one dominant copy of that allele. And 25% of their offspring will not be able to curl their tongue because they only got the recessive trait. So it is possible for two parents who can both curl their tongue to have a baby who cannot curl their tongue. That's shown in the monohybrid cross. The other cross that I show you here is the X-linked cross. That is a sex-linked trait then. Let's talk about that for the example hemophilia, because we have H's here. Hemophilia means that your blood is not able to clot. If you get injured, you will continue bleeding and bleeding and bleeding. That is an X-linked trait. That is an X-linked recessive trait. So in this case, the mother, she does not have hemophilia because she does have that big H, meaning she's healthy. But that little h means she's a carrier of hemophilia. The dad does not have hemophilia. He has X with a big H. That means he's healthy. And then he has a Y chromosome. And we don't attach an H to the Y chromosome because it's an X-linked disorder, not having anything to do with the Y chromosome. So when we cross these parents, this offspring is going to be a female because XX is a female. And since it's big H, this is a female who is completely healthy and is not a carrier for hemophilia. This daughter, again, daughter because it's XX, does not have hemophilia. She's healthy because she has that big H from her dad. But from her mom, she got the little h. So because she has X with a little h, she is actually now a carrier and could pass hemophilia on in the next generation. Now, this is a male, so he will pass on his Y chromosome. This offspring is going to be a son, big X with an H and Y. So when there's a big H there, that means that is a healthy offspring. So healthy male offspring here because it's XY and the X has a big H. Now this boy, again, it's a boy because XY, But this boy, he inherited just a little H from mom. Dad was not able to provide a big H to protect him. And so this little boy with X little h y is going to have hemophilia. Most likely, he would, in olden days, he would pass away at an early age because of the bleeding problem. But nowadays, he would be able to survive because he could go to the hospital and get blood transfusions or stitches, that sort of thing, if he were to become injured. We have modern medicine that could help. But these are two Punnett squares. They can show autosomal traits, ones that are on chromosome pairs 1 through 22, or sex-linked traits, which are on the X or Y 23rd pair chromosome. The last diagram that we kind of mentioned before is pedigrees. These are just diagrams that can show a trait over many different generations. So this would be generation 1, and somebody has a certain trait if they're colored in, generation 2, and generation 3. And you'll notice there are circles and squares. Most often, our circles are going to be the females, and the squares will be the males. And usually, if it's colored in, that means they have the dominant trait, and if it, or sometimes even a recessive trait, but they have a certain trait, and the open ones do not have the trait. So you can kind of trace through a family who had certain traits and who did not. It's a good way for people who are having a career in genetic counseling to be able to talk to their clients about the chances of possibly having an offspring with a genetic disorder. So Punnett squares show combinations for just one genetic cross. Pedigrees show combinations for an entire family. Again, thank you so much for listening.